I was hitting a bottle.
The organisms exceptionally in this place, so I wonder how many people are in the Red Path Museum right now looking for a speaker, but because of department day we decided to move it here, and it's my great pleasure to introduce Dr. Dennis Higgs from the University of Windsor, who, well, I guess his talk, what he says, is Acoustic Ecology, so he's a mix of a neuroethologist, ecologist, and is involved in many different things. So he did his PhD in Texas in a marine science program, but then decided to go to a drier place, Arizona, where he was working on this olfactory development of xenopus was the project there, but then that was too dry, I guess, and he went to join the lab of Art Hopper as a postdoc at the University of Maryland, where he worked with fish and alligators and all kinds of critters, working on their auditory sensitivity, using the auditory brainstem response a lot. And yeah, so some of the collaborators from that time, in particular from the alligator work, Daphne Suarez, he's still collaborating with now, not on alligators anymore, but on cavefish. He went to University of Windsor in 2003 and is still there and is the department head. And so he's a man, obviously, but very successful in his research, part of a strategic research grant minister, and also a recent recipient of a synergy award for work on aquaculture of salmon. So he's the Behavioural person. And for that, I look forward to your talk.
Thank you. Thank you for the invitation. I hadn't been to McGill yet, so I was quite excited to get the invitation from Rudiger and see a couple former students from Windsor. And I did have a very geographical career, so when I teach at Windsor now, they get very confused and scratch their head about where the hell my accent comes from. So if I slip in a y'all now and again, Texas was a positive influence on me. And my research approaches are sort of varied because my students say I have attention deficit problems. I get bored easily, but I'm going to focus on today, I think, one consistent story and sort of the bulk of the approaches I take on fish acoustics, fish sensing, and I sort of cross-camp between neuroscience and ecology. A main focus is sort of on how animals, especially fish, use sensory systems to actually behave in the environment, how that develops and how it actually interacts to help them do what they're supposed to be doing. Before I get into that, I have to step into my department chair role, which is not, some days a good role and it is not, but Windsor gets a lot of grief. It went through a lot of hard times for the economic downturn, both the university and the area. I think now it's turning the corner. It gives me hope again, so Windsor's trying to reinvent itself. Forgive the administrative speak, marketing, branding, Windsor 2.0, I have no idea what the heck that means, but the good things about it is the positive aspects. Windsor, they're pushing this sort of what's cool in Windsor. There's lots of things to do in Windsor. It's actually a really nice area to live. I can afford to live on a small little farm near campus. But the good things academically, they decided to actually invest in faculty again, which is a positive thing. So Windsor right now is, I think, poised to go through a good period of growth and sort of rejuvenation. So as department head, I'm quite happy about that as a faculty member of Windsor. We're actually, in our department this time, we have two positions open that we're currently trying to fill. One, ecosystems ecology, one, cellular molecular neuroscience, and next year I think we're going to have one more position. We'll see if it gets approved by the provost. But I think we're poised now to reinvigorate. When I got hired, there was a nice spate of hiring around my time. So there's a bunch of us in sort of the similar career stage. I'm hoping now across campus they're going to be bringing in a whole new suite of people, so it's a really good place. I think now in science is going to start taking off again. And another ad, for various odd reasons, I've got more funding than I expected. So I am looking for graduate students as well. Scientifically, it's also a good spot to be in. And what I'm going to tell you about today is some of our work at Windsor, some other work in New Zealand that I think all tie together. But scientifically, for an aquatic person like me, I am a marine scientist officially. So I like working in aquatic systems. We're located where the D of Detroit is, across the border, but we're surrounded by lots of different ecosystems. And one of the stories I'm going to tell you about is the local invasive fish in those ecosystems. But as an aquatic person, it's a really nice place to work. I have access to lots of different habitats. We have a very strong aquatic type group that collaborate reasonably well together. The province thinks that the province ends there, the government, but it actually extends down in southwest Ontario. So it's been, I think, a good place for at least me to land, because I can work on lots of different systems. Anything I want to do freshwater, I can do, if I choose to, locally. And one set of stories I'll tell you about is some work we've done in there. But then because I'm marine science, I like to travel as well, so I'm going to tell you some stuff on New Zealand work as well. Before we get to that, when you think of fish, you don't think of ears, you don't think of hearing, you might think electric fish. But fish do have ears, they're just not externally obvious. Often when you think of fish sensory systems, it's not electric, you think of visual systems. Fish don't have external ears. I had to explain to my mother when I got this job. Fish do have ears, however, and in some sense, some levels they work actually as a model. Mammals have semicircular canals. Fish have semicircular canals. In both cases, they're working for balance, not hearing. Mammals have a small saccule and utricle, probably not hearing, and then they have hair cells and a cochlea, which are responsible for sound transduction in the cells therein. Fish, instead of having a cochlea with cells right around it, have three end organs. Utricular, in some species, is a hearing function. Some of the shadow work we've done with ultrasonic maybe. But then I got these two other big end organs, the saccule and lagina, that do drive predominant hearing responses, probably less vestibular. And at the cellular level, both the fish and the human are roughly the same. In both cases, you've got sensory hair cells that are responsible for transduction of sound. The way they work in humans and in fish is those conocilia and microvilli, not real hairs, bend, that transduces neural impulses eventually, that's detection of sound. In us, it works by vibration of that cochlear membrane, causing differential shearing of those hair cells. In fish, it works because those hair cells are overlaid with a dense calcareous otolith, the ear stone, and what transduces that bending, opening of the tip links, making ions rush in or not, is differential movement between the fish and the otolith. The otolith sits on top, fish moves, the otolith doesn't, and you get sensory transduction. So at the cellular level, people do use them as a model for memory and hearing, but for me, I'm more interested in what that means to the fish. Many people that look at fish otoliths just look at them as a way of aging fish. There's a large body of work on fish otoliths that don't worry about hearing. But I care about, as sound impinges on that fish, for today anyway, how does that, A, bend those hair cells, and then how is that processed at the brain level? It gets a little more complicated in fish because essentially the fish are the same density as the water, more or less. As sounds impinge, they move the water molecules, they move the fish. And hearing only happens because that otolith is denser, and that then has differential movement with the hair cells. But the trickier part is actually what sound means underwater. Relatively easy to think about in air as I'm talking to you, there's a pressure wave that go out, hit your eardrum, eventually vibrate your cochlear membrane. In fish, it's more complicated. That's going to be part of the story I'm going to tell today. Because some fish have two pathways for sound to reach the ear, impinge on these hair cells, cause transduction. The direct or centroneural pathway is sort of what I already described. In this case, some sort of sound wave, we'll call it for now, hits the fish, moves the fish, and you get differential shearing. And so you get actual physical movement because the water is physically moving the fish. And essentially that particle displacement is causing bending of the hair cells. All fish that we know of can do that. They all have ears, some look different than others, but they all will be moved by particle motion of sound waves. That then will transduce some neural impulse with the right frequency and cause movement of the hair cells. Some groups of fishes also can do pressure detection. So they have indirect conductive pathway in which sound pressure, not particle motion, will hit some, in this case, the swim bladder. That'll shake the swim bladder, just like a sound wave hitting a balloon will bounce back and forth. These pressure waves will cause swim bladder vibration, and then that, in various ways, gets transduced up to the ear as particle motion. So at the hair cell level, the only way the hair cell works is with particle motion. That's all it can detect. It can't detect pressure. The fish that can detect pressure do it by having some air-filled sock that then shakes the fish and transmits it into particle motion. Keep that in mind. It's going to be important in a second when we think about how fish are using sound and even what we mean or should mean when we talk about fish hearing. There's lots of different ways, well, there's a few different ways to do this conductive pathway. The two most common is the smoke, some overworking line. The ovarian ossicles are what all these stereophytes fishes have. Catfishes, minnows, goldfish have an arrangement which they have a swim bladder there that shakes. There's a series of modified vertebrae, some pectoral elements, but basically a bony chain that then shakes the ear and allows pressure detection. Many fish, when they have to have a swim bladder, we've done some experiments on zebra fish that as they develop that swim bladder and those connections, that's what this is from, I'm clearly saying, their hearing ability essentially improves, bandwidth increases, because they've now got this ability to sense sound pressure. Another major group, the clypeads, herrings and anchovies and their relatives, they have a direct bubble of gas onto a bulla. It ultimately connected to the swim bladder, but instead of this chain of bones, that bulla will vibrate. It's directly connected to the ear, and actually by an elastic connection. As that bulla vibrates in response to sound pressure, that ear can vibrate back and forth. In these cases, what you get is better sensitivity, but also a higher frequency range. And so actually this stuff we've done in the some of the shads can hear ultrasound, so even higher than we can, we think, because of these direct connections. So what the pressure component adds is an increased ability to detect frequency and increased bandwidth in frequencies, and an increased sensitivity. The problem is pressure waves in the sea, or in the lake, in the water, they propagate further than particle motion does. So particle motion drops out fairly fast. This is an idealized unbounded medium with relative sound level. The particle motion component of the sound wave drops quickly. The pressure, and it even goes further in nature, but ideally goes out much further than the particle motion. What this means is, if you're close to the source of sound, you're getting both particle motion and pressure. If you're further from the source of the sound, all that's available to detect is pressure motion. Where this becomes a problem is when we talk about fish hearing and study fish hearing, we almost always do it at areas very close to the source. Either in a loud situation, in which you're well in this near field range. Even in the field, we have difficulty seeing fish further away. We spend much of our time looking at fish responses to sound in this sort of overlapping pressure particle motion range. Despite this, it's getting better now, but almost everybody would report sound levels in terms of pressure. It's difficult to measure directly particle motion. But we say, okay, I'm going to play some sounds, measure fish responses, I'm measuring fish hearing. What we can talk about today is some work that we're looking at that really, and some others are doing similar things, is making us sit back and rethink what we mean by fish hearing, because in this particle motion range, where most of our experimental work is, you're getting both the ears and the lateral line stimulated. So the lateral line has neuromasts. Neuromasts at their basic level are just a bunch of hair cells, same as the ears are. They're in a different setup essentially, but at the cellular level, it's still a hair cell, bends in response to particle motion. So close to the source in this near field range, and the exact value of that distance will vary depending on where you are, but you're really looking at combined responses, at least for some frequencies, of the ears and lateral lines. It's only further from the source that you're really looking at hearing per se. And it's some work I'll tell you about from New Zealand that sort of set me off on this bandwagon to really rethink what we mean when we're looking at response to the sound. And what that really might mean for evolution of hearing or evolution of acoustic ecology of fishes. And we're arguing, by Farber, that much of what fish are really doing in terms of listening to sounds happens probably mostly in this more integrated zone, or it's probably more important closer to the source. And so what we have to think about is how these integrate together and what that all means. What fish are doing with it is still open for debate as well. Why do fish hear? What are they listening for? And sort of the classical answer, or an easy answer, is they're listening to each other. Fish, believe it or not, do make calls. They do sort of sing. And this is just I didn't invent the sound possibly, but kind of the sea trout, some of our round goby stuff. This is a herring, which actually is making calls out of it. I can't swear around the camera. Out of the butt. The herring, FRT, fast repetitive tick, they're actually releasing gas bubbles through the radius. And there's some argument that they're actually using that to communicate in schools. It's still a call. It's any bird people in the room, I'm sorry. It's not a song. Some might never have a call, but it's acoustic utterances by fish. And they do respond to species-specific calls in species-specific manners. So they do respond to each other's calls. They are using that for hearing, but it's probably not why hearing evolved. And it's probably not why hearing evolved in fish. And I say that because, partly because many fish that hear very well, goldfish for example, don't make any calls at all we know of. Lots of fish that do make calls don't hear particularly well. So what we think now, this is not me as many in the field, is instead of evolving just for calls, it's probably evolving for more auditory scene analysis, like humans do in a cocktail party effect. And so if two fish, this is from a review we talked about this book, when they're calling, they're often in very close proximity. There's pressure waves between the fish. There's also particle motion going back and forth between the fish. And this is why we're arguing it's really both ear and lateral line. But they're almost always fairly close together when they're calling and responding to each other. At the same time, though, in nature, that these two idealized fish are calling. You've got other fish calling to make masking noises. You've got lots of environmental noises going on, waves crashing, wind, rain. And so the argument has now been, and we need to work on this still, fish hearing and therefore our hearing evolved not through specific stimuli, but basically to pick out sounds from background noise. And the argument has been made that I don't make sense, but I don't have evidence for yet, is that higher frequency hearing may have evolved because fish need to tune out the low frequency background noises, rocks rolling, waves crashing. And those that evolve high frequency hearing may have evolved it to basically pick up signals from noise better. That, and I'm going to end with sort of that, the evidence we need for that argument. But that's sort of where we think evolutionarily has driven this. But it's a place the field really hasn't done enough work on. That's what I'm going to end my talk with, of where I think we need to go with that regards. The stuff I'll talk about today is two separate sets of experiments on how we actually measure what we call fish hearing. This is common throughout the field. What we're calling neurophys is a relatively simple neurophys. And what we do is we take a fish, put him in some tank somewhere in a different tank setup. You can do speaker in the air, speaker in the water, different debates about it. We essentially play a fish a sound, have electrodes just under the skin, above the brainstem, and basically get a whole field of evoked potential. When we get that, these are traces from, actually this fish. And what you see is here's the increasing sound level. At quieter sounds, you don't see much change. At loud sounds, you see a characteristic change in the brain waveform. And so we trace that back and say, that's auditory threshold, we saw a change in the waveform. It's a little more, better analysis of this, but essentially, the fish heard something. And so what many people do is they put these fish in these tanks, play different tones, and get an audiogram. I'll show you some of these. And it's a quick way to see, does the fish detect the sound we're playing? It doesn't tell you if the fish cares about the sound, if it means anything to the fish, but it says it can detect those sounds in that tank with a speaker underwater. In my lab, we also do behavioral trials. We let the fish tell us, does this sound matter? So standard phonotastic experiments. This one's a round goby, which I'll be talking about later. We've got two speakers, that one's playing, that one's not. I'm going to play a sound. There's a bit of feedback in it, but the little thump, thump, thumps you hear are actually goby calls from the speaker. And if the fish is happy and the sound matters, you get a nice phonotactic response. Those thumps are actually fish calls. It'll go right to that speaker, stay there for a while, and then it will swim away eventually. But it's a good way to tell, does that fish care about the sound? And what I'll show you later is behavioral calls. We can play different calls to that speaker and see what fish are using to judge that. Again, it's in a tank, in a lab, which has, as I'll talk about, some problems with the acoustics of it. But those are sort of the two main ways people in the field, for the most part, have been looking at least sound reception in fish. What I'm going to do today is focus on two sort of separate, but I think related stories, and talk about things where we think there's gaps in this field. It really sort of took off after World War II, when the Navy released, the US Navy especially, released a bunch of classified sounds they had in the water while searching for submarines, turned up the sounds of fish calls. And so the field grew from there, but it's sort of taken a different approach, and myself and my some collaborators have been interested in sort of where else we can take the field, how it might differ. One set of stories is with Craig Radford, who's a professor at University of Auckland at Lee Marine Lab. He and I got some funding together. He comes to Windsor. I go to New Zealand. He, of course, got a much better deal out of it. And some approaches were taken that make at least ourselves rethink how to think about sound. And then the other part two is using a local species with three of my now graduated master's students around goby. I'll tell you why it worked out well for me career-wise. Looking at what's that mean ecologically? Is sound an honest signal? There's been lots of honest signaling work in other systems, other contexts. We wanted to know, can it actually work for fish acoustics to actually tell females anything about the males? And I'll start out with the New Zealand stuff, and a couple different studies, but at least in my head they all tie together to tell a consistent story. And so what we did, this is the Lee Marine Lab, it's out of the Marine Reserve. The seasons will switch, so when I'm there at Christmas, I can skate back to my life in a snowstorm. And it's a really nice setup. They have a very nice physiology lab. They've got a long history with John Montgomery as the director there. And Craig and I started working, and we started eventually wanted to look at how do the ears and lateral lines really work together for sound detection. We devised a set of experiments using these three species. The goldfish, again, is sort of the lab rat of fish acoustics. It's a good control. We know it hears well, so it's a good comparator. This guy, the triple fin, is a local species. It has no swim bladder. As near as we can tell, it can't detect pressure waves. It's a good baseline species for it. If a fish can't hear very well, it's probably going to hear like a triple fin does, and probably just part of evolution. And the first reason I went down there with this guy, the big eye. It's a New Zealand endemic species and a couple other species in Indonesia, but the aspersus is largely endemic to New Zealand. And Craig and I first explored a collaboration because of some stuff he discovered with the big eye that made it was a completely unexplored, because much of fish sanctuary work had been in, especially acoustic work, had been in the northern hemisphere. And he brought me down and we got excited about this species because of what it does in an ecological context, because we thought this would be a good model. And the reason we thought it would be a good model is because what they do is they live in caves during the day. They're marine caverns that are completely connected to the open ocean, but they spend all the time during the day in these caverns, and they only come out at night to feed. And they stay in these relatively large schools, both in the cavern and at night. We think they're avoiding predation by hiding in the caverns and come out at night to feed. But what Craig's group discovered is while they're out feeding, they're making little clicks to each other. They make very characteristic sounds. And his group is arguing that they're using these as basically contact calls, like a zebrafinch, and not as annoying. Sorry. But they're making these contact calls, and he got interested in what are they hearing when they're making their calls. His student was interested in sound production. But he and I got together and looked at reception because his student was kind of mucking around one day in the lab, just sort of wanted to see what the inside of the fish looked like. He did some very careful, very nice dissections. And this is the ear down here. There's the swim bladder here. He didn't have any ovarian oscles or anything, but he noticed this weird ligament at the time he didn't know what it was. That goes right from the otic capsule to the body wall of the fish. I looked at that and immediately thought, hearing specialization. You've got a swim bladder right next to the ear. That swim bladder shakes, you should shake the ear. You've got this tight ligament, which at the time we didn't know what it was. Directly connecting actually looked like, in these gross dissections, that it was actually hitting the swim bladder, was hitting the ear, and also connected to the outside body wall. And where it connected to, there was a large group of neuromasts at that connection. So I immediately thought there's got to be hearing specialization. Craig works with folks in Auckland who had a spare MRI kicking around that do better anatomical reconstruction, and he knows all the details of this one. Well, you see the swim bladder is very large, and in front of it is thicker than the back of it, so it's got a nice thickening in front. And this ligament turned out to be a known ligament in fish, bottom-lobe ligament. What it's supposed to be is a structural ligament. In all other work we found, it connects the pectoral girdle to the side of the body for structural support. But in these ones, the only ones we found so far, it actually directly connects the ear to the swim bladder to the outside of the body where there's a big bunch of lateral line neuromasts impinging. So we immediately thought this could well be a brand new hearing specialization to allow pressure detection in big eye. And so we did a bunch of experiments to test that. We did selective ablation and then tested the hearing before and after ablation to figure out what's going on. And so we took a big eye, anesthetized it, did a hearing test on it, still alive, took it out, and we did a couple manipulations. We either cut that ligament with a fine pair of forceps, went in and cut it, and we did both bilateral and unilateral dissections. We popped the swim bladder, which is a common technique to see if the pressure detection is going on. But then we also wanted to know about the neuromouse. We filled the lateral line canal with a plastic elastomer to block activation, we hoped, of the neuromouse themselves. And then we ran hearing tests before and after manipulation. That's the control, so test the fish twice. You don't get much threshold difference over a range of frequencies, so it's good. You test the fish twice and get the same response. If you pop the swim bladder, you do see an increase in threshold. They hear less well. Again, not unexpected. If you cut that ligament, though, you see a much bigger hearing deficit. So ligament was playing a role in transmitting sounds to the ear. And so this and some other evidence told us this was a new hearing specialization. Then also, if you fill the lateral line, do nothing else to it, but just fill the lateral line, hearing itself also changes. Again, it's in a tank, in a lab. So it led us to believe that A, the ligament is a specialization, but also B, the neuromasts are playing a role in sound detection. It bothered us at first because it's supposed to be hearing. So that sort of set us off in this path of wondering about what's going on with the neuromasts, and are they linked? And we argued that they were, and people seem to believe us. And so we wanted to explore that in some other species to see really how much of this linkage occurs. Is it just a weird big eye thing, or what's really going on? And so then we designed a set of experiments, same three fishes again, trying to separate particle motion from pressure. It's hard to do in an underwater stimulus. If you're playing a speaker, you get in both modes. So the first thing we did was, this is our standard physiology setup. This is the New Zealand one we had transported around. We basically have a speaker in a tank, fish here, electrodes plugged in. That again has both pressure and particle information in it. So then we designed a mini shaker table, a low-budget shaker table, in which the fish sit here, out of the water. There's no speaker, so the sound stimulus just runs through this rotary shaker, and we can do it in different directions. So we can have pure particle motion detection, or a mix of pressure and particle motion against these species with these different sorts of hearing specializations to get at which part of hearing is really hearing, pressure detection, which part might be just pure particle motion detection, and how that might differ between these species. When we look at this, again, that's the goldfish. Goldfish are sort of the champion hearers. They do pressure and particle. We now know big eye doing pressure and particle. That one has no modification for pressure detection. And if we look underwater with a speaker, we see that, lo and behold, down is better hearing. Goldfish hear better. Big eye aren't bad. A little triple fin doesn't hear as well. So with a traditional speaker setup, you do see differences in sound detection. But if you look at just the particle motion component, there's absolutely no difference. So there's no specializations in the ear itself for enhanced hearing. It does all come down to something about those extra pressure sensitivities. And we're arguing evolutionarily, probably all the changes are on the specialization side. A fish ear is a fish ear is a fish ear. But we wanted to then separate really what role is the neuromass playing in all this stuff in something like a goldfish. Because when you do these low potential recordings, they're relatively gross scale. We're getting basically a whole brainstem response. And this is the auditory nerve, those are lateral line nerves. We put electrode roughly there, electrode roughly there. A lot of people have done this, and you get your response. What we're worried about is we're probably actually picking up all of our units. We're not picking up an auditory response. You can do that with single or multi-unit recording, but what many people are doing is just brainstem. And brainstem, there's lots of overlap, they're probably both integrating and torus. And so traditionally when people have been using physiology to study fish hearing, they're getting both. And you can knock out neuromass selectively with various drugs. This is in a goldfish. That's a control. We did knock out the neuromass to prove we did it. That's the little dots of neuromasses. When we do that, hearing does change. So when you just knock out the lateral line, you're getting a change in hearing. So the controls, we don't see, again we tested them twice, no threshold shift. The scrape, if we physically scrape off superficial neuromasts, which doesn't have any effect, but the canal neuromass, what's in the lateral line canal, if you knock those out, you do get a decrease in hearing, especially at lower frequency range, which is telling us that what we're calling fish hearing is not fish hearing at all. It's hearing plus mechanoreception driving those responses. And people have been sort of doing this stuff, and they called it all hearing. We're now arguing you can't, and people are starting to believe us. We think evolutionarily, at the evolution of high frequency hearing, it probably is this indirect pressure pathway. And by using a set of techniques, we can sort of not worry about, there's not much changing the ear itself, but more in this indirect pathway. But also because that most sound communication occurs over relatively short distances, you got to be careful what you're calling hearing, what you're not calling hearing. And if you're looking for modifications for acoustic communication, you really have to look at modifications both in the auditory pathway and the mechanosensory pathway. And thinking about how fish communicate, it's important to think about the spatial scales at which they're doing it. The second story brings me back to Canada. And that, because I like to build these for bad reasons, does it all matter? This was all lab stuff. We can see that they communicate differently. We can see that they respond. But what does it really mean to the fish? Can we bring it back into